great pleasure to welcome Dr. Peter Franks. As he's probably waiting for me to say, he is one of the best cooks I have met since I came. His homemade pasta is fantastic. Also, another little-known fact about Peter is, and I looked at his very distinguished resume, is that he did once upon a time work on birds like myself. He actually worked on brood parasitism, the effect of cowbirds on sparrows. See, I know more about you than most people. Since that time, he has become a biological oceanographer and a professor in the integrative oceanography division here at Scripps Institution of Oceanography. He's been here for nearly a decade, and in that time he's developed a very high reputation as a world-class researcher, and he's also a very, very good speaker, so you should have a wonderful time this morning, and he's also one of our very best teachers. His research focuses on the interaction of physical and biological processes in the plankton and encompasses such visually striking and economically relevant phenomena as toxic algal blooms and red tides, as well as less obvious patchiness of the phytoplankton on the scale of millimeters to hundreds of kilometers. And today, Peter is going to speak on new tools, new discoveries, thin layers of phytoplankton in the sea. Thank you, Peter.
Thank you. Wow, that's a lot to live up to. Wow, what a great introduction. I'm not sure it's all true, but I'll accept it. Why not? So today I've been asked to speak about some of my research, but because this is also going to be part of the Centennial Lecture Series, I've been asked to give a historical perspective on my research. All right, this title of my talk, New Tools, New Discoveries, Thin Layers of Phytoplankton in the Ocean. I'm going to give you a 100-year history of this sort of research here at Scripps. First of all, the theme of the talk is microscale phytoplankton patchiness. And just to get some definitions out of the way, in case you're not all oceanographers, by microscale, I mean scales that are less than 1 meter. So, you know, this sort of scale. These are the scales that the organisms live their lives in the ocean, most of the phytoplankton and zooplankton. By patchiness, I'm referring to the spatial scales over which we see big changes in concentration of the organisms. And I'm not defining big and I'm not defining scale here because it's a very complicated subject. But when you think of a patch, think of walking across a lawn and then all of a sudden you see a bush. That would be like a patch. And this is the sort of thing that we're looking for in the ocean. And finally, phytoplankton. These are the single-celled photosynthetic organisms in the ocean. They're bacteria and protozoa. They fix organic carbon through photosynthesis. Now, when we think of phytoplankton, we mostly think of things like this, the diatoms. These are single-celled organisms covered in a shell of silica, or the dinoflagellates, which are single-celled organisms covered with a very beautiful shell made of cellulose. And up until 1980, these were the phytoplankton in the ocean. And around 1980, we discovered that cyanobacteria, Synechococcus and Prochlorococcus, are actually far more abundant and more important in terms of primary production than the diatoms and the dinoflagellates. Now, these are bacteria. These are single-celled protozoa with nuclei. I don't want to mislead you that these are all the same size. This is more what they would look like when drawn to scale, and you can see that the bacteria, I couldn't actually make them quite small enough there because of the pixel resolution of the system, but they're very numerous and very important. Just as an example, this room I pasted off the other day is about 1,000 cubic meters. So if this were 1 cubic millimeter, so 1 millimeter by millimeter by millimeter, a millimeter is about the thickness of a dime. It would contain about 100 or 1,000 of these bacteria, and each would be the size of about an M&M. And it would contain perhaps one of these large phytoplankton and maybe 5 to 100 phytoplankton about the size of a golf ball or a ping pong ball. So you can imagine with 1,000 M&Ms just hovering around in here and a few ping pong balls and maybe one big blob that would be about the size of me or perhaps a blue whale, it would be pretty easy to walk around in here and not bump into a phytoplankton cell. And this is the sort of environment that the zooplankton are foraging in when they're looking for food. And so this is the type of environment that I'm exploring and trying to understand how it's structured, the physical and biological mechanisms that lead to changes in the distribution patterns of these organisms. So why would we even care? Well, first of all, phytoplankton and zooplankton interact on the microscale. If you want to eat another organism, you have to get up to it and chomp it. And so their foraging scales are centimeters, millimeters, even microns. And if you want to mate and have sex, of course, you have to find the other organism. Most of them have contact mating. They don't broadcast spawn. And so you have to actually find a mate, have contact with them, exchange genetic information. This is an interesting reason. The patchiness of food resources. Up until recently, we've done most of our sampling in the ocean using nets and bottles, and I'll describe a lot about that shortly. When you draw a net from, say, 100 meters depth to the surface to get an idea of how much food is out there, you're taking an integrated sample over that depth. And what we found is that the average food concentration in that net is not enough to support the growth of most of the organisms that we see there. But clearly the organisms are growing. So the conclusion is that the food has to be patchy. It's distributed in blobs that somehow these organisms are finding and exploiting to keep their growth rates up. And so we know the patchiness is out there, we just don't know what it's like, where it is, why it's there. And then finally, the big question that I'm heading for in the remainder of my career and perhaps several lifetimes afterward is, why are there so many species of phytoplankton? There's been a lot of work on how they can coexist and problems with competition and that sort of thing. My question's more fundamental. How did they get there in the first place? And I'll talk a bit about this down the road. All right, so a century of sampling phytoplankton patchiness at Scripps. This is going to be a very Scripps-ocentric view of sampling. There's lots of good work that's been done lots of other places. I just don't have time. So, measuring phytoplankton patchiness has always been limited by technology. That's still true today. Every time we develop a new instrument, we learn new things. 100 years ago, there was no way of actually looking at the organisms in the ocean, in situ. We had to get them back to the lab somehow and count them, measure them, do experiments on them, and try and infer patchiness from these samples. And the two mechanisms that were used for getting organisms back into the lab or onto the ship were bottles and nets, and that's still true today pretty much. The first bottle I found that was developed here at Scripps, or at least associated with Scripps, was developed by a fellow named Charles Kofoid. He was the associate director of the Scripps Institution of Oceanography before it was actually named that. He was very instrumental in the inception of Scripps and in the discussions of the creation of Scripps at the Del Coronado Boathouse back in 1903. He was very famous for his studies of dinoflagellates, and he developed this thing called the Kofoid bucket. And it was a bucket that would descend in an open position. Here's the bucket itself. There's the top lid and the bottom lid. Then a messenger, this weight, would be sent down a wire. It would trigger a mechanism at the top, and the bucket would fall down and close. And then this little spigot would be attached at the bottom, and the sample would be drawn out. And it's not clear to me why he published it with panel A to the right of panel B, but that's the way it was in the paper. The next bucket I came across was developed by this fellow, a very dapper guy named Winfred Emery Allen. He was an assistant professor here at Scripps from 1919 to 1943. He was an assistant professor that whole time because he never finished his PhD, like many of my classmates, actually. He worked on it and worked on it and worked on it. But he developed or acquired a 20-year time series of phytoplankton samples from the end of the Scripps Pier. These were, I believe, weekly samples over 20 years. Can you imagine? 20 years, every week sampling at the end of that pier. It's astounding. There's been nothing like it since, as far as I know. And he developed the Allen bottle, and you can see it here being deployed by Fleming, a famous oceanographer. It's, again, an open-closing bottle. The bottle goes down open so the water flushes through it. You close it at depth with a messenger that was sent down the wire, and then the sample's drained out, and here it's being filtered through a net. This net was made of silk bolting cloth, which was fairly unreliable, and at the bottom, the cod end, we call it, is a little jar that would capture the sample. And so here are two bottles that are named after two famous Scripps scientists. You might think, wow, that's really great. These guys developed bottles named after them. I went into the NOAA archives and discovered about 100 bottles developed between 1890 and 1940 that were named after scientists, mostly Scandinavians and Germans. And it seemed that any scientist, any oceanographer worth his salt developed a bottle and had it named after him. So it wasn't really that big a deal. All right. So what did Allen find after 20 years of exploring phytoplankton off Scripps? Well, here are some quotes from his papers. He found extreme differences in abundances at individual stations along any line. And so this is fairly definitive that there's patchiness in the horizontal direction. But what about vertically? He said catches from levels below 50 meters were rarely equal or superior in size to some at higher levels. And then, just in another paper, he had a longer quote that contradicted that. Sometimes many diatoms appear at lower levels, though few at the surface. Sometimes many appear at the surface when numbers are small below. And sometimes rather large numbers may be found at all 7 of the levels sampled at a particular station. This is the state of the art of our understanding of patchiness back in 1940. But he also noted that we'd sometimes find these cloud-like aggregations of diatoms and dinoflagellates. And so you can see from here 7 of the levels sampled. So he had 7 samples vertically, and they were probably about 10 meters apart. And this is the finest resolution they had at the time for estimating patchiness of phytoplankton. So this is the state of the art by the beginning of the Second World War. And then along comes this fellow Herbert Graham, who was working here at Scripps in 1943, published a paper. He said, the time-honored method of counting the number of plant cells is not satisfactory. First, it's a slow and tedious procedure. Second, the results must be expressed in numbers of cells when size of the cells in different species of organisms is very different. Furthermore, silk bolting cloth, which is usually used for collecting the samples on board ship, is not a reliable filter. And there have been lots of studies since that prove that very definitively. So what Graham suggested was that instead of bringing the samples back and counting the phytoplankton under a microscope, perhaps we could get some measure of the amount of phytoplankton without actually having to count the individuals. And he was suggesting using chlorophyll, because chlorophyll A, or a derivative of that, is present in all photosynthetic organisms. And so it's representative of the phytoplankton, but not the zooplankton. So he presented a method for extracting chlorophyll A from phytoplankton filtered onto a chemical filter paper. This is a much finer filter than silk bolting cloth would have been, and it probably actually collected the cyanobacteria, although they didn't exist back then as far as he knew. And then he measured the absorption of the chlorophyll. So the chlorophyll was extracted using acetone in this case. He extracted the chlorophyll and then measured the absorption of red light in a spectrophotometer. And this is very accurate, but it's not very sensitive. And so you need a lot of chlorophyll to measure the decrease in light as it's passing through this sample of chlorophyll. So they had to filter large amounts of water, usually 4 liters or so. So that was the state of the art for about 20 years. And then along came Osman Hom Hansen, who's still here at Scripps, Carl Lorenzen, Robert Holmes, and John Strickland of the Food Chain Research Group. In 1965, they published a paper where instead of looking at the absorption of light by the chlorophyll, he looked at the fluorescence of light by chlorophyll. So what's fluorescence? Well, here's a chlorophyll molecule. It has a long chain of carbon that anchors it in the protein matrix within the chloroplast. And it's got a series of carbon rings here that hold right in the middle the magnesium ion. When you shine green light on this structure, a lot of it will be absorbed and used for photosynthesis, popping electrons around and doing all kinds of good things. But some of that light will actually, the energy will be lost, but not all of it, and the light will be re-radiated as red light at a lower wavelength. And the amount of fluorescence, this is called the fluorescence now, so the green light has come in, it comes back as red light. This fluorescence is proportional to the amount of chlorophyll A. And if the amount of chlorophyll A is proportional to the size of the cell, then the fluorescence is a pretty good measure, somewhat imperfect, of the biomass, the amount of phytoplankton that's present in your sample. Now, fluorescence is a very sensitive measure because instead of looking for the disappearance of light, you're looking at light being made. So you can filter out the green light and you'll have a black field, and then all you'll see is the red light coming from the phytoplankton. And this is a very sensitive measure. It's like looking at stars, or in a sample where you've extracted the chlorophyll, you'll see a nice red glow from it. So they extracted the chlorophyll and used this fluorescence method. Then Carl Lorenzen the next year published a paper where he said, well, forget all this extraction stuff. Why don't we just look at the phytoplankton themselves? So he took tubes of living phytoplankton and put them in a clunky instrument, something like this. This is an old Turner Model 110 fluorometer. It's got doors and knobs and bells and whistles, and they're very complicated to use. But you can put the sample in, shine a blue or a green light on it, and measure the amount of red light that's fluoresced back. But he went one step further. So this is now living phytoplankton. He said, well, What if we take a Turner fluorometer like this? The sample would normally go in here. You can still see these fluorometers in use today down in my building. And instead of just having a tube go in here, what if we plumb it and have garden hose pumping water through here? So we'll bring water in at the bow of the ship, we'll put it through a garden hose, pump it through the sample chamber and then out again, and we'll record the voltage, the fluorescence that comes back. Then we can steam our ship along and measure fluorescence along this transect and get a continuous measure of the patchiness of the fluorescence. And he recorded all this on a big piece of chart paper. And this is a very short paper. It's about 6 pages long, and it had a couple of plots in it. But it was such a fundamentally new idea. It was like the atomic bomb in terms of phytoplankton ecology, because now we have a way of measuring the patchiness of phytoplankton without having to bring samples back on board. So this was huge. And that's basically what we do today, except now we don't actually have to use these clunky big Turner fluorometers with garden hose. We have, let me give one more point here. At the end of his paper, he had this one sentence: the instrument could also be used for obtaining vertical profiles in conjunction with a pump. And this sounds kind of silly, but this is actually how I did my thesis work. I had a Turner Designs fluorometer, 40 meters of garden hose, a pool pump all plugged in, and I just pumped water from 40 meters depth and very slowly raised the bottom of the hose up and got a vertical profile of fluorescence. The alternative, which was the state of the art back in the 1980s, was to take the whole fluorometer, which is about this big, put it in a giant pressure case about the size of a garbage can, put a pump inside the pressure case to pump water through the fluorometer, have a big electric cable going down the wire, and lower the whole kit and caboodle all the way down and do a vertical profile that way. And that was the way to do it 20 years ago. And now we have beautiful instruments like this. This is a Chelsea fluorometer. It's about the size of a couple of cans of peas or canned tomatoes or something like that. Most of the new ones are about the size of a soda can, these tiny little fluorometers. You can put them on any sort of package. They take very little power, and you can do vertical profiles with these instruments and get a measure of the fluorescence on the way down. They're sensitive from about 0.1 to 100 micrograms of chlorophyll per liter. I'll tell you a little bit more about that shortly. And their vertical resolution is really about 1 meter, perhaps as small as 20 centimeters, but that's rare. So we're still just barely getting into this microscale realm. 1 meter is just where the microscale begins. So we're not quite there yet, but we're doing much better than we were. So what do you see when you actually measure fluorescence? Well, almost everywhere in the ocean, you see a profile like this. The black line is the fluorescence versus depth. Here's depth from 0 down to 80 meters. This profile was taken just off San Diego in the summer of 2001. This is the temperature profile with a strong thermocline, a decrease in temperature over a very short vertical distance here and here. This is a very strong density gradient which will stop vertical mixing from the surface. But you always see, pretty much always see, this subsurface fluorescence maximum, which is also usually a subsurface chlorophyll maximum. We call it the deep chlorophyll maximum, or DCM. And even though most of you have never seen this, and I've never actually looked at it myself, I've just looked at traces like this, it's probably the most widespread biological feature on this planet. It's in every ocean almost all the time. It's way down just where the light reaches about 1% of its surface intensity, and it's just an incredibly robust feature. And I'm not going to say anything about how it gets there, because surprisingly we still don't really know that much about what controls it. Clearly light is very important, and I'll leave it at that. Now the chlorophyll concentrations. Off San Diego, the average chlorophyll concentration is about 1 microgram per liter off the end of the pier. That makes for very blue-gray water. It's very clear water. Here the maximum chlorophyll concentration was about 0.65 micrograms per liter. The water will get colored at about 20 micrograms per liter. This would be a red tide when the phytoplankton bloom enough that they actually color the water. And you've probably seen some of our really dense red tides when the water goes kind of a brick red or purple color. That'll get up to 200 to 500 micrograms per liter. This is really, really dense, soupy phytoplankton. Not pretty stuff. You can't really see through this. All right, so let's get back to the microscale. So we've done about half a century now of patchiness sampling, and now we have to leave Scripps and go somewhere else to find the microscale stuff that's been done. The first paper I could come across was one by Darren Bock et al., his colleagues from Scandinavia. in 1979, where they took an in situ fluorometer, I don't know what kind, I think it was built in-house, and they lowered it down, this is 0 to 55 meters, they lowered it down in the Baltic. Here's the temperature trace, and here's the fluorescence trace. What you can see is below the thermocline, you have these huge spikes in fluorescence. And these spikes are about a meter thick or less, and they can be many times the background concentration. This is actually a log scale for chlorophyll up here. And they suggested that these peaks in fluorescence were actually thin layers of phytoplankton, big pancakes that would be floating around there, that formed as the phytoplankton sank down, and then they hit a little density gradient and they slowed down and accumulated, just like a traffic jam on the road. The cars are spread far apart where everybody's driving fast. When there's an accident, everyone slows down to look and the cars get closer together, and then they get far apart again. So the phytoplankton are thought to just be moving through this. I'm somewhat skeptical of that explanation, but unfortunately there's no way to test it right now. The next example isn't until 1993 when Tim Coles of Oregon State University developed a really amazing laser fluorometer. And this is a beautiful instrument. It's shaped like a giant torpedo. It's about 2 meters long, and it falls nose first down through the water very, very slowly. And it can sample the fluorescence about every 2 centimeters. Here's the temperature trace between 30 and 60 meters, and here's the fluorescence. And this is a particularly special instrument. It can gather a spectrum of fluorescence. So it doesn't just look at one wavelength of light. It looks at, I think there's about 20 or maybe even 100 wavelengths. And because different phytoplankton have different pigments, they'll have different fluorescence properties. And you could actually distinguish among different taxa using the spectral properties of the fluorescence. But this is the fluorescence of chlorophyll A. And again, you can see these great big spikes occasionally. And over here we have a measure of the turbulent mixing. Where this line is low, turbulent mixing will be very low. Where this line is very high, turbulent mixing will be quite strong. And you can see that occasionally these peaks will line up with regions of very low turbulent mixing, which gives some suggestion that there's an interaction here between the physics and the biology in the water column. And again, these layers, or these peaks, tend to be less than a meter thick, so on the order of 30 to 60 centimeters. And he didn't really interpret them beyond showing that they existed. There again was some suggestion that they might be layers, but we don't really know that because all we have is a sample that's about the diameter of this pen falling down. This is not a pen, this is a laser pointer, falling down through the water column. And so you can't tell from one sample whether this is a layer or a blob or anything else. And so here's where we are. We still really have no resolution on the scale of the organism's ambits. We don't know how the phytoplankton are arranged in space. We do know that there's variability out there. And so these peaks that we see, are they layers? Are they just little blobs that we've happened to run our sampler through? Or are they some chunk of something that got jammed in the instrument as it went down and then popped out again a few seconds later, making something that looked like a layer but in fact wasn't? And that's certainly a possibility. What is the spatial arrangement of the phytoplankton within these features? And in particular in the deep chlorophyll maximum, how does the community structure change as we go from the surface waters into this big feature? It would be like walking from a desert into a lush forest and then out again. Surely there's going to be changes in the community. And then back to my old question, why are there so many phytoplankton species? So I'll give you the answers to all these questions shortly. All right, now we finally get to the end of the century of sampling phytoplankton patchiness at Scripps, and the pinnacle of evolution, myself and my colleague Jules Jaffe here, and we developed an instrument called the laser imaging fluorometer. So the laser imaging fluorometer is still a fluorometer, but instead of sampling in a small sample volume as water flows through, this was Jules' suggestion. Why not use a big sheet of laser light, a very thin sheet, 6.5 millimeters thick, shaped into a flat plane. We'll use this sheet of green light to stimulate the fluorescence of phytoplankton, and then we'll image the fluorescence in this plane with a very sensitive CCD camera. So we're shining green light on the phytoplankton to stimulate fluorescence, then we have filters on the camera that take away all the green light and only see the red light. So we'll only see the sources of fluorescence. So we're only seeing photosynthetic organisms. This is a big deal. We're not seeing the zooplankton, we're not seeing dead material unless it was dead recently enough that it's still fluorescing. The imaging area was 32 by 32 centimeters, so that's bigger than a regular sheet of paper. And we had 320 micron resolution there. A micron, there's 1,000 microns in a millimeter, so 320 microns is a third of a millimeter. So the pixel size, these pixels are about 2 millimeters by 2 millimeters, so they're smaller than these pixels that you can see on the screen here, quite a bit smaller actually. And the initial results were good and bad. We certainly got a signal, this is good. The bad part was the whole instrument was mounted on a wire that was attached to the ship. And as the ship moved up and down, the instrument moved up and down, and every time the shutter of the camera was open, we got a nice smear of all the phytoplankton through the camera plane. And we had to reject about 3/4 of our images because they were smeared because of the ship heave. So how do you get away from ship heave? Well, you make an instrument that you can kick off the ship and it'll fall just by itself. And so that's what we did, and Jules named this Phyto-Phy. I think just to make me say it so often. This is a free-fall imaging device for observing phytoplankton. And yes, I know. But we actually do use phy. It's a nice shorthand for phytoplankton. Although, of course, any other country would say phi and phytoplankton. But there you go. So here's the instrument. It's an autonomous package. It's battery powered. You might be able to see these packages here are the batteries. There's buoyancy up at the top, and these big fins keep it oriented. There's a tube here which contains the camera, and the computers and sensors for tilt and roll and depth, the pressure sensors. There's a laser over here which shines this flat sheet of laser light, and the camera is here. Now the sheet of laser light is angled at 45 degrees below the instrument so that the camera can image at 90 degrees. And the imaging plane, shown here, is about 80 centimeters below the whole system. So as the system falls, it's imaging water below it, and this water, we've shown to ourselves, is undisturbed by the instrument itself. And this is very important when you're looking at microscale features, because you don't want to be stirring up the water that you're trying to explore. And we built a scale model of this whole thing, and we put it in the flume down at the Hydro Lab, and we showed quite conclusively that there was just no influence of the flow of the instrument on the water at all in the imaging plane. So we're pretty confident that what we're seeing is what's really there. And so this instrument was ballasted. This is a huge instrument, by the way. It's not very pretty, but it's effective. It's 2 meters across by 3 meters tall. It weighs about 2,500 pounds. And it's so beautifully designed by Fred Ullman, our engineer, that the difference of a pound or two of weight on it will determine whether it sinks or floats. And so it was just beautifully ballasted. We could ballast it to fall anywhere between 3 and 10 centimeters a second, depending on what we felt like doing. And when it reached a predetermined depth, it would drop a ballast, and it would come very slowly back up to the surface. And then we'd capture it with the rings up here and bring it back on board ship. So let's see. We did a bunch of deployments of this instrument in the summer of 2001, and I'm just going to describe one here because it would be too long to show all the data that we gathered. And this is a particularly nice one. It occurred just as the sun was setting, and the drop of the Phyto-Phy was flanked by 2 drops of a CTD rosette system. Now CTD stands for conductivity, temperature, and depth. This is an instrument that measures a vertical profile of conductivity, which is like salinity, and temperature, which I hope you all know, and depth. And from that you can calculate a vertical profile of density, and the density is really important in determining how the water flows and how much mixing there might be and all these physical properties of the ocean. There is also a Chelsea fluorometer on it. This is not actually our package, it's a picture I stole off the web, but it looks a lot like this. So there's a regular fluorometer on it. There is a CTEC transmissometer, which you can ignore for now. And then you can see this rosette. This is an array of bottles. These are opening, closing bottles that are a lot more expensive than Allen's bottle or Kofoid's bucket, but they do exactly the same thing. They can be tripped electronically at any depth. There's a little console up on the ship. You just push a button, you got your sample. You bring the samples back, and we did exactly what they did back in the 40s. We filtered it for chlorophyll A, and we preserved samples for doing cell counts. So nothing much has changed in 100 years. Then we did a drop of our Phyto-Phy. It was falling at 10 centimeters per second, and we gathered 313 images, about 1 every 2 seconds. And then we did another cast of the CTD rosette. So we have lots of real information flanking our brand new instrument to see if it's actually working. So the first question we had was, here we have an instrument, it probably costs several $1,000,000 to build. It's hard to tell, really. It weighs 2,500 pounds. It's really big. Does it work as well as a soda can-sized fluorometer? Well, here's the results from our soda can-sized fluorometer. This is the vertical trace of fluorescence. The black squares are the extracted chlorophyll that we obtained from our bottle cast. And so the fluorometer is working pretty well. This is good. And that was the cast before the phytophy drop. Here's the cast after the phytophy drop with its extracted chlorophyll, and they both agree with each other pretty well as we might expect. So what about the phytophy? Well, how do you compare the two? What we did was take each image from the phytophy and we averaged the fluorescence within an image. And so we're imaging a volume that's 32 by 32 centimeters and 6.5 millimeters thick. So it's quite a large volume compared to a regular fluorometer. And so we just averaged the fluorescence and plotted that versus depth. And here's what you get. Yay! It works really well. It's obviously very sensitive at these extremely low chlorophyll concentrations, and we can see that it shows the same shape as the regular fluorometer. Now, anytime you do a fluorescence cast, and I'm sure most of you have done this, you'll find these spikes and peaks in your profiles. And for the first few times you do it, you scratch your head and you say, gee, I wonder what that was. And after a while, you just ignore them. But now we can tell you what these are because we can actually take a picture of these sorts of features and find out what they are. And this is the big exciting thing with this new instrument. So I'm going to show you an animation of some of the images from this drop. I'm going to show you an animation from 0 to about 58 meters. There are 3 panels on this plot. This left-hand panel is depth and chlorophyll concentration. So this is the average fluorescence from our phytophy instrument, the plot that I just showed you. You'll see a little red ball here. This bouncing ball will indicate the depth from which the image was taken. The image is shown here. This is a section from the larger image, and it's a section where we're pretty confident about the data that we obtain. The image area is 13 by 24 centimeters, so this is quite a bit larger than real life. And you'll see a lot of these white streaks at the beginning. I'll describe it as the thing's running. And then this panel shows the fluorescence intensity, where 0 is very low fluorescence and white colors are very intense fluorescence. So let's start this running. The first thing I want you to notice is what happens when we go through this little peak. Now, each of these little dots is a phytoplankton cell, as it turns out. Wow, wasn't that wild? So each of these dots is a phytoplankton cell, and we did considerable work in the lab to prove that each of these is an individual cell. We can actually see cells that are a lot smaller than the resolution of the camera system. Remember, the camera is 320 by 320 microns. We can see cells at least as small as 5 microns and perhaps smaller. Now you can see they're all streaky. This is because in the very low density waters near the surface, it was falling a little bit fast. But once it hits the thermocline, right about here, it slows down a bit, and now the streakiness is completely gone, and we're seeing these very clear, well, relatively clear images of these cells. And you can see here the phytoplankton arranged in space. Occasionally you see these really large blobs that go through. These are probably aggregates of phytoplankton, and some of them could be the guts of zooplankton. Now we're going to come down into the fluorescence maximum, and this is just an amazing series of images, I think. You can see the background here is very black, and you see a lot of white stars on top of it, the fluorescence of these individual cells. What you're going to see as we come into the fluorescence maximum is the background is going to start to get lighter and lighter as the number of really small cells increases very suddenly. We don't resolve these cells very well, and there's so many of them, we'll find many within a pixel. Wasn't that amazing? And that all happened in the space of about a meter, so between my waist and the floor. The chlorophyll concentration went from pretty much nothing up to its maximum value. You can also see a lot more of these really large cells. These are big diatom chains, aggregates of phytoplankton. These are really huge cells. They'll be on the order of 2 or 3 millimeters in size, so you could very easily see these with your eyes. And as we go down, you'll see the types of cells change. If you've watched this animation as many times as I have, you actually get an appreciation for who's there and where they are. And I'll show you some of this shortly. Now we're getting toward the bottom of that fluorescence maximum, and you're going to see the background fluorescence is going to drop away, but you'll still be left with a lot of these very large fluorescent particles, the phytoplankton. There we are. Now the background is fading away. And it still looks like there's a lot of material here, but the fluorescence is extraordinarily low. It's about 0.1 or less micrograms per liter. So while it looks like there's a lot of material here, if you were to add it all together and put it up in one corner, it would account for very little of the actual fluorescence. Most of the fluorescence is actually in the background where it looks black. It's very low values of fluorescence, but because there's so much of it, it accounts for a lot of the total fluorescence. Now we're coming down into a very interesting layer, and you'll see a lot of really large, robust cells coming up, these sorts of things. They're sort of long and chunky, and it's a layer, I think, of perhaps copepod fecal pellets or aggregates of diatoms that have begun to sink through, and I'll show you a bit more about that shortly, but it's a very distinct layer. Now the animation is going to repeat if everything goes well, and we'll go back to the beginning so you can see that layer again, just because it's such a spectacular thing to see. Here we go. We're back up at the top, coming down through that layer, and you'll see a very sudden increase. Boom. And then it's gone. It's quite amazing. All right, so let's explore some of these features. First of all, let's look at some of the layers, that top layer that we see in the image. So here again we have 3 panels. The left panel is fluorescence versus depth. Now we're going from 0 to 50 meters. And I've put a little gray bar over this region of the vertical profile, and it's a little less than a meter thick. And I expanded that here. This is 70 centimeters worth of depth, and there are 3 images in this 70 centimeters of depth. And you can see that the layer is pretty much fully contained in this one image. You can see that there's really no hint of the layer in this image and none in that image, and you can see the layer actually falls off at the bottom of this image. So the layer was probably only about 20 centimeters thick. And coincidentally, well, not totally coincidentally, we popped a bottle in this layer in the previous CTD cast, and when we looked at that bottle sample, we found it was full of this organism, a dinoflagellate called Lingulidinium polyedrum. And Lingulidinium is one of our favorite little red tide dinoflagellates around here. And 2 weeks prior to our cruise, we'd had a very dense and very beautiful bioluminescent display from Lingulidinium polyedrum. At the time of our cruise, the water was fairly clear. The bloom had ceased, and there was a lot of feeding by salps and other filter feeders to clean the water out. But there were still enough cells left to make this sort of layer. Now, how would they do that? Well, dinoflagellates have 2 flagellae. There's one that runs around this groove and another that comes out that groove. They can swim, and we know they have certain behaviors. We don't know exactly what controls their behavior, but it's quite likely that for some reason they decided they all wanted to hang out right on this density surface, and so they swam there from the top and from the bottom, forming this very dense layer. And the concentrations of cells within this layer were about 10 to 20 times the background concentration. And I'll show you how we know that very shortly. And this layer persisted for at least 1 hour, and it was probably there much longer because we saw it in a series of casts that we made that day. All right, now the other thing you can do, which has never been done before as far as I know, is use these images to do continuous cell counts through the water column. Now you can imagine taking a whole bunch of bottle samples, say every 30 centimeters over 80 or 90 meters depth, and then counting all the phytoplankton in them. And imagine how many years that would take you. Well, now we can do it in a matter of minutes using computer algorithms, a program called MATLAB. We can develop algorithms for analyzing our images, and we can quantify the area of individual fluorescent dots. And then you can choose any size of particle and count them up, or any shape of particle if you want. And so what we did here is picked cells that were greater than 5 pixels in area. So these are really big cells, greater than a millimeter. They're really visible to the naked eye, huge things in terms of phytoplankton. And then we counted them up in every single image as we went down through the profile. So this is pretty exciting. You've never seen anything like this as far as I know. Here's your fluorescence profile. It's the same one that I've been showing you. But now here, the red line is the number of cells per liter for these very large cells. So here we have a continuous count of cells through 80 meters of the water column. And what you can see is a huge peak in the cell concentration right in that dinoflagellate layer. was about 3,000 cells per liter. And we can just zip this right off the image. It's quite exciting. But now you can see these large cells, the red line, have quite different structures than the regular fluorescence, the chlorophyll biomass. The chlorophyll has quite sharp gradients spatially, but the large cells have much stronger gradients than the bulk chlorophyll. And you can see that these cells are arranged into 3 quite distinct peaks through the deep chlorophyll maximum layer. And then there's this funny layer deeper down. So let's take a look at some of these peaks. First, the upper one. I'll show you an image from the trough above that layer and then from the peak of that layer. And you can see a very different character. These are just the largest cells now. I've taken away all the background fluorescence, and the increase in concentration of cells is quite apparent. And these cells are fairly small. You see the occasional long, this is probably a diatom chain, but mostly they're small dotty looking things. If we look down at the next layer, we can see the trough and then the layer itself. In the trough, you can see an increase of about a factor of 2 of the number of particles. The trough is somewhat uninteresting, but then in the layer, you start to see some of these more robust looking diatoms. These, I think they're probably diatoms anyway from our bottle samples, rather large phytoplankton. The next layer is quite interesting. The trough above it and then the layer itself, rather than those robust looking ones, you see these long, slender looking diatom chains, and it's very curious that they're all oriented in the same direction. This is not because the image is streaky, it's probably because the center buoyancy of the diatom in these long chains is a little bit different than its center of rotation, and if there's any shear in the water column, these diatoms will all be tilted slightly and they all line up, and this has actually been seen before in the ocean. But you'll see how the shape is quite different. You don't see these long, slender ones so much in the previous layers, and you don't really even see them, I think, in this layer. You certainly have chains, but I think, personally, they're quite different looking. And of course, we'll have to get bottle samples to verify them, or get finer resolution on the camera so we can actually look at species. Now, finally, this bottom layer is a very interesting one, where you see these big, robust, chunky-looking guys. They may be fecal pellets that have sunk out of the deep chlorophyll maximum from zooplankton grazing, or they could be aggregates of diatoms that are suddenly sinking out, and this layer may just get deeper and deeper. And I haven't looked at the casts that we did after this to see if that layer was actually migrating down as it was sinking out. That would be something to do. So far, we've learned that the Phytophy, this laser imaging fluorometer, gives the same information as a regular off-the-shelf little soda can-sized fluorometer, so this is good. But it can also show whether fluorescence peaks are due to a layer of cells or one big bright blob that happen to go through the instrument. And we often see both cases, layers of cells and bright blobs in our data. We can quantify the spatial gradients of chlorophyll over scales as small as a millimeter, and this has just never been done before. We can actually look at these images and see what's happening within an image. You can often see gradients just over the space of one image. We can perform cell counts centimeter by centimeter through the water column, which has never been done before. And the instrument clearly reveals that we have these thin layers of large cells throughout the deep chlorophyll maximum and in other regions of the water column as well. So how might these layers form? Well, there are 4 ideas that I could come up with. The first one is that maybe they're just growing there. For some reason, at that layer, the environment is perfect for this type of cell, and they grow faster than they're being grazed, and their concentration increases. So the suggestion would be that there's some environmental optimum right there, and the cells, this particular species, just grows and proliferates. Another mechanism is that the cells may accumulate at that layer. So the dinoflagellates that we saw are probably accumulating there. They probably swam there and just stayed when they got there. Why they decided to end up there, it might be like a singles bar or something like that. I don't know what the signal is, but there they were. Another mechanism was suggested by Darren Bock and Astheimer in their paper, that the cells just sink out and gradually accumulate on this layer and then maybe fall through a bit later. Or they could be doing the opposite. They could be floating up from below and accumulating in that layer. And the next mechanism is one that I published on a few years ago, the vertical shearing of an existing patch. And my wife persuaded me to steal my daughter's Slinky for this demonstration. So the idea is that this is a patch of phytoplankton, and if you take a sample vertically here, you won't see anything. If you take a sample vertically here, you'll see a big patch this thick. If you take a sample over here, you don't find anything. But now the water starts to shear this patch in this direction. Now, I wish I had another hand, but if you take a sample through this, you'll find quite a thin layer. And the more it's sheared, the thinner that layer will get. So you can, start with an existing patch and then shear it just as waters move over each other and stretch it out to form a thin layer in any given location. And if you have multiple patches and you start to shear them out, you'll get multiple layers. And this has nothing to do with excessive growth in a very thin layer or swimming into a thin layer. It's just that you started with some big patch and made it much, much thinner by shearing it out. Hope that's very evocative. And finally, photoadaptation. There's been a lot of discussion as to whether these layers really exist or whether the deep chlorophyll maximum is even a maximum of phytoplankton biomass. Because when cells are deep down in the dark, just like the plants on land, they'll form more chlorophyll. And so you can get more fluorescence per cell, even though there aren't more cells. So if you just have a fluorometer and you drop it down, you could see a peak in fluorescence without actually having a peak in chlorophyll or a peak in biomass. But what we've showed, at least here off San Diego, is that there's definitely a peak of biomass there, and you can actually count up the cells. There's more of them in the deep chlorophyll maximum. So even though photoadaptation is probably occurring, it doesn't account for all the features that we see. All right, well, a couple of years ago I got promoted to full professor here. No need to congratulate me. It was an accident, I'm sure. But one of the nice things about being a full professor is that you're really hard to fire. And so I can say fairly ridiculous things, and I'm much harder to get rid of than I used to be. And so I'm going to do that now. And so why are there so many phytoplankton species? So my question is, how do they form? On land, if you want to make a new species, you take a population, you separate it from its congeners, you take two populations, and you put a mountain range between them, or you put a river between them, or a road, or a desert, or you put them on islands, or something like that. And gradually, genetic drift and other sorts of effects will allow them to drift apart genetically until they're separate species. They can no longer come back and exchange genetic information in a viable way. How do you do that in the ocean? It's all connected. It's all mixing around. How do you make a new species? If you start with one phytoplankton species and it grows really well, why isn't there just one phytoplankton species exchanging information with itself all over the planet? But here's my speculation. Most phytoplankton species have many genotypes, just like humans do. I look different than Jeff. We have a lot of the same genes, but some of the genes are different. Jeff might like cold weather more than I do, and so we'll have different environmental optima. It's the same with the phytoplankton. These genotypes may have different environmental optima, so they might form layers in different places or at different times. Now, why would that matter? Well, most phytoplankton reproduce by binary fission. This is easy. You don't have to exchange genetic material, you just divide. So you can build your population up to quite a high level without ever having sex. And then they might have sex just once in a season, once a year. Sounds kind of boring, but some of them apparently don't have sex at all. And so they have sex only infrequently, and the sexual phase might not last very long. Now, Ginger Armbrust published a couple of papers. Ginger is up at University of Washington. She was actually a classmate of mine at the Woods Hole Oceanographic Institution. And she showed that there's a lot of variability in the sperm-egg recognition proteins in diatoms and chlorophytes in the ocean. And if the sperm can't recognize the egg, you're not going to be able to exchange genetic information. So this is a very strong barrier to sexual exchange. So when you put all these things together, you have a spatial separation of genotypes. Perhaps they form a thin layer at 30 meters, and another genotype forms a thin layer at 31 meters. But they don't see each other. And then they have a sexual phase where they all start to mate with whoever's nearest by. But if you're a meter away, that's 100 miles in terms of phytoplankton. And so this spatial separation, it might only be a meter or so in the ocean, but when you combine that with a short sexual phase, you can have total genetic separation of the populations and allow speciation even with no mountain ranges or deserts or anything else in the ocean. That a distance of mere meters might be enough to allow speciation. This is total speculation, and if anyone wants to come back to me on that and discuss it further, I'd appreciate it, especially you at home. All right, just to finish up. This was a section from an image that we took on our first cruise in the summer of 2001, and it was just so neat I thought I'd better show it. This is a section, just a small piece from a larger image that we took at the deep chlorophyll maximum, And it was during the bloom of a bunch of diatoms, Bacteriastrum, Guinardia, and Pseudonychia, which is a potentially toxic diatom that we occasionally find locally. And it was actually floating at about 35 meters below a red tide of Lingulodinium polyedrum, and we found it sitting there for at least 3 weeks. And the thing that's striking is this one diatom chain, this is a Pseudonychia, you can tell by its sort of curved aspect, is almost 2 centimeters from tip to tip. That's as long as the tip of your finger. Well, it's longer than the tip of my finger. But it's huge. These are enormous cells, and you can see them throughout this image, these great long diatom chains. Now, we don't always find these diatom chains in our samples because when you put them in the bottle and then run them out the spigot, they break up. And we often find a lot shorter diatom chains, but I think these sorts of images are giving us a much better idea of how the subsurface chlorophyll maximum is structured. What are the phytoplankton doing down there? How are they arranged relative to each other? How far apart are they? What's the potential for competition? What are the physical forces that are structuring this environment? And these are the sorts of things that we want to get to in our future work. We'd like to put more physical instruments on this free-falling platform so that we can measure the physical flows and the microscale physical structures at the same time that we measure the biology, so that we can really get some insights into the dynamics that structure this ecosystem. We've already begun to quantify the scales of patchiness on the scales of the organism's ambits, on the scales that they live their daily lives. We can calculate things like nearest neighbor distances. How far apart are these cells? How much potential is there for competing or interacting with each other? We could actually just count up the areas of these cells and estimate phytoplankton carbon, which is a very difficult measure to make because when you take a sample from the ocean, it's full of phytoplankton and zooplankton and dead stuff. How do you separate them to get just the phytoplankton carbon? This is not a trivial problem. And finally, we're looking at measuring the actual flows of the water through a technique called particle image velocimetry. And by following the motions of particles over very short periods of time, basically taking 3 snapshots very quickly, we can follow the tracks of the organisms and look at the turbulent structures in the water column and see how the phytoplankton relate to these turbulent flows. All right, finally I'd like to acknowledge a lot of people who have helped me a great deal in this talk. Jules Jaffe, scientist, engineer here at Scripps, has been instrumental in, well, he built the instrument and came up with the idea. He and I have interacted a great deal. His student Dave Zawada did a lot of work on the optics. Nando Simonet, who works in his lab, and all the wonderful people who work in his lab have been a great deal of help in this. At the aquarium, Cheryl Peach, who invited me to give this talk, has been very generous with her help, and Miriam Pulsino did some scanning for me of some of those old documents. Then Deborah Day and Carolyn Rainey at the SIO archives were extremely helpful. You should all go and visit them. It's an incredible resource. And finally, this work was funded by the Office of Naval Research and 2 divisions of NSF. So thank you very much for your attention. Are these related to acoustic scattering layers? Yes, in a very interesting and dynamic way. We actually published a paper on that a few years ago. The acoustic scattering layers are much larger organisms, zooplankton, that migrate up and down. And we actually recorded the acoustic scattering at the same time we measured the fluorescence, and we found that one of the big peaks in fluorescence was just a desert to the acoustic scatterers, the zooplankton. They migrated up right through it and then stopped at a different level. And this is just fascinating. We have no idea why they avoided it, but the acoustics wouldn't really scatter off these small phytoplankton unless you have a really super high frequency. So it could be an acoustic scattering layer, but you won't see it in your fish finder. After I get them, how long do these creatures live? Well, if you put them in formalin, probably less than a second, because I usually preserve them to look at them under the microscope. So I don't care whether they're alive or not. I'm just counting them.
Thank you very much, Peter.
My pleasure.